Well, good evening. Welcome, everyone. This is the American Shoulder and Elbow Society, one of our research webinars. This is our first of a series of three that we'll carry out over the next year. I'm Scott Rodeo from Special Surgery in New York City and joined by my co-chair, Peter Simon, chairs of the ASCS Research Committee. The first title tonight, we're going to explore randomized clinical trials. We think it's well established that often considered the gold standard of research trials, but they're challenging and there's real pearls. So we're going to go through some of the pearls and pitfalls of running these types of trials. Our prior slide, if you go back one, I think number two, well, here we go. This is our panel of experts here. Connie Chu from Stanford University, Eric Wagner from Emory, and Kurt Spindler from Cleveland Clinic, all of whom have a lot of expertise in the area of clinical trials. Our next slide just briefly shows our disclosures here. And the fourth slide here, we are going to go through these three important areas. Here's click through these, please, Steve. We're going to have each speaker address these three general areas. Number one, planning and funding these randomized trials and clinical trials. How do you execute a study? And number three, study completion. And how do you really bring this across the finish line and get to the point of publication? So before we start, I'd like to thank Smith & Nephew for their generous support of this webinar. You can go ahead and run that. And we'll go right in with our speakers in a moment here.
Good evening, everyone, and welcome to the ASCS Foundation webinar series. I'm Scott Gunn, Vice President of Shoulder Marketing and Sales at Smith & Nephew. As the sole sponsors of this series, we're thrilled to have the opportunity to introduce you to Smith & Nephew and our innovative product portfolio. For over 100 years, Smith & Nephew has been at the forefront of medical technology, committed to improving lives through our pioneering solutions. We're excited to share our latest advancements in shoulder arthroplasty and sports medicine. Our team of experts have been focused on providing you options throughout your patient's continuum of care with cutting-edge products like Regeniten, a bioinductive implant that redefines biologic healing for rotator cuff tendon growth, and the elegant ATOS shoulder system, which embodies stability, simplicity, and flexibility. ATOS is a streamlined system for both anatomic and reverse procedures, allowing for an elevated operating room experience. Throughout this webinar series, you'll hear from leading experts in the field who will discuss the latest research, clinical evidence, and best practices in shoulder and elbow medicine. Thank you for joining today, and we look forward to sharing our passion for innovation and helping you provide the best possible care for your patients.
All right, well, thank you. Again, we thank Smith & Nephew for their support. We'll start with Dr. Constance Chu. Constance is Professor of Orthopedic Surgery at Stanford University with extensive experience in running clinical trials. So we'll jump right in. Connie, thank you.
Great. Thank you, Scott and Peter, and everyone for joining us tonight. I'll be talking a little bit in a very conversational manner on some of the pearls and pitfalls of more than 25 years of doing clinical studies and clinical trials. And so really, research is fun, it's exciting, and it also is a lot of work. And so I'm going to really start by emphasizing how important it is that you're clear about the research question that you're trying to answer. And for us surgeons, it usually boils down to an unsolved clinical problem. And it will evolve over time as your research evolves. And I'll just share with you that my goal at this point in my career is to advance public health by improving joint health and mobility. And the way that I've chosen to do that, I don't have commercial conflicts, but my research has been funded for the past 25 years by a variety of federal grants. And these grants have centered around the concept of early diagnosis and early treatment to prevent or delay the onset of osteoarthritis. And so at the present time, I'm working on 9 active projects, and they're part of 2 program project grants, and I've highlighted the clinical trials. There are 4 clinical trials, and the 5th clinical trial is actually in horses as a gene therapy trial. And so really, once you have your burning clinical question, I think the most important thing in starting out is building a committed team. And we'll go through some of the elements. If you're looking at a surgical procedure or some other clinical treatment, it's really important to look into your own practice as to whether you have the patient population that's needed. Most of the time, to really answer an important question, you'll need to collaborate. And so it's very important to have partners with the desired patients and also shared research goals. And on the left here, I'm going to list a few pearls. You'll also want to look for collaborators, and these can often be scientific collaborators, whether they're clinical or basic science collaborators or translational researchers that have key expertise that you need to get your project done. If you're doing a clinical trial, early involvement with a biostatistician is critically important. And then going into the different assessment technologies, I use a lot of MR imaging, and so I have at this point a long list of MRI collaborators. There's also a very important person on your team, and that is a clinical coordinator. Early in my career, I didn't have one, and it's something that you'll see come up again and again. That clinical coordinator is indispensable in conducting not just clinical research, but particularly if you're looking at doing a clinical trial. And you're going to need money to do this. And the most common sources are some form of self or practice-related funding, industry funding, and federal grant funding, and usually it's some combination of all three. Now, on the right-hand side, I'll just list a few potential pitfalls, and insufficient resources comes to the top of my list. And really, the top one is time. Good research and clinical trials and clinical research take a lot of time, and it will take a lot of personal time on the part of the person that's the most interested in driving this. And the second you'll need is, we've touched on it, and it's one of our top topics tonight, is money. And with money, you can buy expertise, but it's still very, very important to build that committed team. You want everybody who's working with you to be working towards this common goal because it's really that teamwork that's going to carry you through. The second pitfall I want to comment on is underestimating the scope of the project. And these are mistakes that I've made, all of them. Generally not the same one twice, but sometimes it's been a little too similar for me to be happy about. And I think it's that patient recruitment, and you can have the patient population, and I myself will see the primary patient populations I'm recruiting in my own clinic, but that doesn't mean that I have a recruitment pathway, which means while I'm trying to take care of the patient clinically, how do we get that person funneled into the research process? And that really is where having that clinical coordinator will make all the difference, along with other factors that will be individual to each particular study. Now, if you are doing a clinical trial, typically there is an intervention that you're looking at. If it's a medication trial, it is really important to do a safety and dosing study, even if you're already funded and that wasn't part of the grant proposal that got funded. If you want your trial to be effective, you want to look at what you're using for your therapeutic intervention, and take that extra time to look at the specific dosing that's going to give you the therapeutic effect that you're measuring. And so many of the NIH uh, 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 trials grants will have that process baked in as the first phase of that grant application. And it is for this reason. I'm not the only person that's made this mistake. And so those things are put in so that going forward, people can do better about it. And it all boils down to having enough prior sweat equity, just working with that team so that the clinical trial you're about to embark on, you can demonstrate feasibility and know that you can get it done. And really the third pitfall, which comes up time and time again, is unclear objectives. And so this is something you can be very clear about your research question, and you can be very clear in your mind of what the procedures and how everybody's supposed to interact. But it's really important that the entire team is clear on those objectives. And it actually takes repetition, repetition, and repetition. And so once you have a multidisciplinary team together, in a committed team, these are just some of the elements that are on my team at this point in my career, where we're doing multidisciplinary clinical outcomes assessments on the same patient population. Really, it starts with your base team to look at the clinical problem. And it's usually your partners or other people in your specialty society that will comprise that team. And I think you'll hear more about that in this webinar. The next topic is study execution, and I just put that clinical coordinator back right at the top of the list. That individual or individuals, depending on the size and scope of the trial, is absolutely essential. The oversight, yes, your clinical coordinators and other people on your team are going to certainly have responsibility for implementing some aspects of the oversight, but as the principal investigator, it's absolutely critical to put in your personal time consistently, regularly, and without fail. That's critically important. On clinical trials, you can have FDA inspections, FDA regulations, certainly data safety and monitoring board that is external to your group, and also your institutional review board oversight, and they can conduct spot inspections. You really want to have everything in airtight, that everything's going according to the plans, the procedures, and it's being monitored. Now, recruitment and retention is key. And this actually, a lot of clinical trials fall on the recruitment and retention. And I've said before, even if it's a population where you walk out of clinic and said, oh, I saw 5 people that were potentially candidates, but someone's got to stop in that clinic and make sure that they get into the research pathway. They have to screen them. And in many instances, you'll end up with 5 potentials. You might end up with 1 or 2 that are actually committed and able to go through the entire study. And the recruitment really needs to be monitored continuously to make sure you're staying on track and within the parameters of your grant. Sometimes you'll have to expand the indications. And establishing relationships with everybody on the team, but also the patient that's being recruited, absolutely critical to get the longitudinal follow-up that you're going to need. And not doing these things end up being the potential pitfalls where sloppy administration or lack of oversight, and then failing to stay on top of your recruitment targets really compromise the study. And you do need to be aware of potential competing studies. And in longitudinal studies, patients may relocate, and so having a plan in place, how to maintain contact with those patients and get the longitudinal follow-up that you need, critically important. And these are all areas that are important to have a plan ahead of time and continuously adjust your plan as reality intrudes on your clinical trial. There will always be something unexpected. And I've had the good fortune of having done 3 clinical trials in horses, and the interventions that we looked at in those clinical trials, in the Goodrich study, Lori Goodrich study, that's being used, the autologous platelet-enriched fibrin glue in clinical practice in horses. Big difference. It's hard enough when you can just make sure that your subjects are captive in their stalls, coming out and doing the procedures that they need to do at set periods of time. But humans and patients are far removed from that type of controlled environment, and so critically important to stay on top of things. And finally, in terms of study completion and publication, this really is potentially the fun part. The primary manuscript is critically important. So this is whatever you proposed in your study protocol, which really for a randomized controlled trial, particularly FDA trial, or an NIH or DoD funded trial, needs to be completed to the letter of how you proposed it, with modifications approved by your program officer and a funding agency. And then you do publish those outcomes according to statement of work. Now, out of that, often years of work, it's the related manuscripts that really are pearls, the sub-analyses, the alternative analyses, and you've now put in the time and you have a well-curated cohort with outcomes that becomes a convenience cohort. So residents, fellows, or students can do these sub-analyses, alternative analyses, and really publish outstanding papers. So this is part of the reward. And it's critically important after you complete a study to do an after-action review. Something I learned in the military, well, it's fresh in your mind. What went wrong, what went well, and how could you have done things better? And it'll help you the next time around. There are many potential pitfalls. Authorship can be one. It's critical to have that discussion ahead of time. Sometimes you have to make additions, and sometimes things don't go according to the discussion, but in general, whatever the plan that was laid out in the initial discussion, in general, you want to follow through on that authorship plan, barring some extraordinarily terrible circumstances. The other potential pitfall is peer review. Sometimes, particularly with longitudinal studies with 2-year follow-up, it can be a few years from when the protocol was approved, and maybe somebody else published a paper and everybody's sitting there going, well, why didn't you look at this? And you're sitting there going, well, that wasn't part of our approved protocol, which had 10 other things that we needed to look at. And really, I think these things come up, but in general, editors and reviewers with a good explanation and appropriate modification of the paper to address that specific concern, you can generally get that across. And then grant closeouts, if your admin up to this point was good, you should be in good shape really to make an impressive report to the funding agency. And so a lot can come out from a team effort over a long period of time. And one of my early goals was to be able to see this invisible cartilage damage that I was convinced was present early after ACL injury. And so team 42 manuscripts later, we're able to see and define pre-OA in these patients. And so that really, there's a lot of joy along the way with all the hard work. And in the discussion, we'll probably talk about this, and I've arrayed kind of a progression in approaching clinical research. And thank you for your attention. And again, it's all about your team.
Wonderful. Connie, thank you. It's a great overview. I thought we'd go through our 3 talks and we'll have some time for discussion, and we can pick up some of these points that will be raised here. So our next talk is Kurt Spindler. Kurt, probably well known to everyone here. Kurt has a wealth of expertise in this area. Kurt was at Vanderbilt for a long time and now is at Cleveland Clinic, Florida. And Kurt is really the primary organizer and really founder of the well-established Moon Group, the Multi-Center Orthopedic Outcomes Network, which is published in the knee and the shoulder. So Kurt brings a wealth of experience in clinical trials. So Kurt, thank you.
It's an honor to be here, and it's a pleasure to talk. And I certainly agree with Connie's outstanding talk before. I like it to crawl and run, crawl, walk, run, fly. I think that's so true. I haven't seen that before. My disclosures. My disclosures have been shown before. So what I did was I just tried to take a little check sheet program, because basically there's a checklist that you have to do, particularly on a randomized trial in a cohort. And otherwise, if you don't do the checklist and you miss some of these things, it can derail it. And so first thing is you need a biostatistician. You're planning and funding, you absolutely need someone as a core member of your team. Don't enlist them afterwards, enlist them in the beginning. That's a tip. Second of all, you must really look at the inclusion exclusion patients at each site. Enrollment is the hardest part of any study, and you have to get it right. And no matter how much you plan, there's something going to happen that's going to make your life difficult. And finally, when it comes to preliminary data, you need to have something that suggests a signal, particularly if you're going to do a multi-center study, because you certainly have to have enough evidence to support a very expensive study to do it in a multi-center way. Pitfalls, I think one of the pitfalls is you decide to design the study, and then you try to engage your team. And that's like putting a paper in front of someone and say, sign off on it. People want to be involved in it. The way to do that is you can design the concept and engage your team in all parts of the way so they feel they've had modification authorship. Second thing is to seek funding for this is difficult. If you expect to get it on your first try for funding, don't try, because basically it's going to take you multiple attempts and multiple avenues to get it done. And I think that really, you really should only do multi-center after you've shown you have an effect in the single center study. So you shouldn't do a multi-center study unless you, in a highly controlled environment, you show you have some signal that works. And now the question is, can you reproduce it? Is the same effect the same in Cleveland or Florida or New York or California? So this is what we did. We did a lot of planning before we did our Bear Moon trial. And this is showing you, I'm showing you by age, because we had to know what ages to do. And this is enough. We tracked 5 sites, 10 surgeons, half a year, prospectively, 510 ACLs. And you can see we have a good population, 18 to 40 years old, which is what our core population is. And then the next problem was we had to get him to the OR within 50 days. That is not as easy as you think, but we have a huge segment of folks, 112 of them that got in less than 50 days, but we got 43, or a fair number, almost half, that were over 50 days. And so that was something for us to optimize. And so when you look at that 510 ACLs, and you take out the non-primaries, you take out people that are 17 and under and 41 and over, and you take out 50 days, that 510 drops down to 112, or 22%. Then that's not the ones that are going to accept randomization, because that's going to be less than that. So it's amazing how your numbers drop out really quickly. And so you really have to over plan for what you think and have some backups and additional sites. Study execution, I agree 100% with what Connie said. I think that when you're trying to pick sites, the most important factor is they have to have patients at the site. If they don't have enough patients and multiple surgeons, the site, no matter how engaged the investigator is, they just won't be able to grab the numbers. And every site is a cost, every site is an expense, every site is something else you have to track. And second, are they trustworthy? Trustworthy? Are they going to do what they say they're going to do? And if they have multiple competing trials, can they enroll in the trial that everyone else is doing? And finally, when you're going to initiate a site, you really have to perform a site initiation visit. You have to go to the site, either in person, preferably, or virtually, to say, let's run through the whole protocol. Let's run through all the research coordinators. Let's run through the physicians. Let's run through X-ray. Let's run through everything that you need to make sure you can get it done. And whatever you think IRB and legal is going to take, it's going to be longer. Unfortunately, I have not found a way to speed that process up. So you have to realize that's going to take a longer time period. The pitfalls that you can get into is you have to track enrollment from the first month and every month. You have to track every site and every enrollment, because it's just like anything in life. If you're not paying attention to it, it's going to fall by the wayside. Finally, funding. You can't leave it to your admins to manage your grant, because all of a sudden at the end, your money's gone and your grant's not gone. You need to meet as a PI with your admin person, because you're always it's always going to take longer than you think. So you're always trying to look for a no-cost extension. You're always trying to look at your money and spend it and stretch it out for an additional year so you can finish up. And I think if you don't monitor a site or monitor yourself, and there's not some monitoring, failure to monitor it is a mistake. All of a sudden at the end of the trial, there's a bunch of missingness and data that all could have been taken care of early on. So in our prospective cohort that we've been collecting at Cleveland Clinic, we delivered it 119,000 times, and we've been successful at administering it 110. So we basically have been able to capture 91% of our patient-reported outcomes, and surgeons are able to capture 94%. That only happens if they're being monitored on a regular basis. And it only happens if they're being monitored and it's part of their metric for success. It wouldn't happen if we didn't monitor it. And in fact, during COVID, we slid, because basically it's hard to monitor people in COVID when everyone's worried about much more important things in life. And then when COVID ended, we got back to monitoring it. And if I were to show you by year, you would see that the compliance and everything else dropped down, as you would expect. And then finally, if we don't monitor follow-up, which we did, so in our follow-up, 84,000 different knee, hips, and shoulders, our follow-up is 75% at 1 year. And that's monitored very carefully. Now, the important thing to note here is the green bar and the blue bar. The green bar is an active person calling them, research assistants. The blue bar is what you get when you have my chart message, email, text messaging, and whatever other electronic. We've never been able to get good follow-up with any of those means before. And then finally, we had everyone outside of it's coming to shoulder. I think total shoulders are going to be pretty close by, but our colleagues that do hips and knees have to report out 50% at 1 year of inpatient total joints. It will be outpatient in 3 years. So we monitor every site every month really carefully to make sure they collect what they need to collect. And I can tell you that the green bar looks good. It looks good in green now. It didn't look good in green when we started in July. It took a lot of gnashing at the teeth, a lot of different things to get the nurses engaged, and we got quality involved in that too. And it's really a problem, because if the system doesn't work, our system in Cleveland Clinic would lose $18 million across our whole enterprise if we didn't do it, but it'll work. But again, it only works because people are monitored and people are held accountable, and there's a number to shoot by. So what about completion and publication? I think that you have an executive team, and I think that executive team needs to meet almost weekly, or at least 2 or 3 times a month. If you set it up weekly, you all know that'll happen 2 or 3 times a month, which is fine. I think you need to meet with the investigators and the coordinators once a month. And the investigators can be a separate call, but the coordinators can also be a separate call as ever managing the coordinators. And sometimes they intermix, but you have to keep them engaged. I think early on, you want to publish a design paper, an iterator paper, a systematic review or something. You want to get some early wins for the team so they feel like they're already part of something. And I think funding wise, again, you're shooting for a no cost extension because no matter how hard you plan, something will come up and do it. And the scary thing is that it's something like 3 out of 4 NIH funded randomized trials never met enrollment. That's a lot of money, right? And I can tell you everything. I've either run a trial or been part of a trial. I can tell you we struggle with enrollment every single time, no matter how we plan. Authorship, the pitfall is not discussing it before you enroll. When you're enrolling, everyone should know where they can be on the authorship. And that's why you want to plan the grant and you want to have them being a collaborator in planning, because that's part of being an authorship. Authorship is part of the design, not just contributing patients, and also part of you want to engage them when you have the statistical output. You want to have a meeting with them, maybe your monthly meeting, and go over the statistics with them so they can vet it out and do it. And I think publishing, I think you really have to, there are, as Connie really worded it superbly, there's a primary outcome, and then there's a bunch of good secondary outcomes, and they're separate papers. And what you want to do is spread the authorship around. You can use a corporate authorship, but basically, if you're the PI, you shouldn't be first author on more than one paper. You should always have someone else trying to write the paper, do the paper, get your whole team engaged in it. And finally, implementation is a real problem. You have to realize that industry has a bias because an industry is supposed to sell product, and they may or may not accept your results. And if they accept it, they'll tout it. If they don't accept it, they won't tout it. And there's some examples we have here. So in the 20 people in the Moon group, no one has left the group in 23 years. I don't know why, but they haven't. They still get on a call the second Monday at 7 a.m. They still call their patients for follow-up, and no paper gets published without unanimous consensus. Who am I to say that Bob Marks doesn't know what he's doing on the paper? He may be wrong, or Chris Kading, or wherever else. And when I was asked to say, what's the number one achievement, and give a talk, I asked everyone in the group, and they felt the number one achievement was really the teamwork of success, that the team stayed together, worked together. And a funny little story is there was one paper that I thought should be 2 papers. We disagreed on a phone call, so we pulled it to a blinded vote. And the blinded vote was 14-2, and I was in the 2, that it should be 1 paper. So we went back and did it as 1 paper. And when they read the 1 paper, you know what they said? It belongs to be 2 papers. It's too much. So actually, it really wasn't good enough for 2 papers. We published 1 and not it. So you really have to realize the group makes a decision, not an individual involved in that. And then finally, every study has limitations. And so in Moon, we cannot evaluate. If you look at if you understand the data carefully, we know football, basketball, and soccer. We can't tell you other sports because we don't have enough of an end really to be able to prove that across. We don't have sample size. We can tell you revisions are bad. We don't have enough revisions to be able to say what are the risk factors, and that's why Mars was born. Mars put together. Rick Wright ran it. 1,200, and a lot of people participated. 1,200 revisions in 2 years that eclipsed the entire published literature on revisions. So teamwork works. And then finally, implementation. I don't know about you, but I don't like Dr. Google, people coming in. So we decided to spend $50,000, have the results written for the 6th grade level, design a website, paid someone to do it, and to monitor. So we put all of our data on there so we can refer to our patients to the website to answer questions and everything else. And so none of this, as Connie so aptly said, it requires a lot of different funding sources. Here's early grants early on, and things that we supported, and certainly my partners, Jed Kuhn and Eric McCarty, we actually supported with our own money early on when I was at Vanderbilt. And again, most of the big funding is the NIH. We couldn't do without their support. So I look forward to your questions, and thank you for the opportunity.
Great, thank you, Kurt. I'm glad to hear you talk about the challenges with enrollment. I lie awake, I think, every night wondering why are we lagging behind in our enrollment? So that's a principal challenge we can talk a little bit more about. So last talk here, maybe we'll have a little time for discussion. Hopefully, Eric Wagner from Emory University School of Medicine will give us his insights on these same general topics of both the pearls and pitfalls and some of the challenges in clinical trials.
Fantastic. It's my honor, and I appreciate this opportunity. My talk's gonna be more from the perspective of somebody relatively early in the career, and to give you a background for those of you who don't know me or aren't familiar. At this point, I've published 16 randomized prospective trials in the first sort of decade of my career, and then we have another maybe 10 ongoing that we are doing both from a single and multi-center standpoint. So I want to give a addition or maybe a different perspective or a younger perspective for how to conduct these, some pitfalls, and kind of why we do these. Real quick, I'm just going to sort of talk for those who are not Constance or Kurt, that are in the earlier phase of actually starting to crawl, maybe even starting to walk, why we do these. Obviously, limitations is an important one. Retrospectively, it's very hard to account for these come factors when you're designing your trials. You want to keep this in mind. Bias, this is something clearly that enrollment's an issue, whether you're doing a single or multi-center study. Keeping this in mind, things like this G-power can help you to sort of calculate. And then fragility is something that I think a lot of studies suffer from, not necessarily the Moon trial, not necessarily some of the one that Connie was mentioning, but a lot of trials that we do with different colleagues in the ASCS. Changing one outcome means that you can change the whole message of the paper, whole message of the trial. And this is something to kind of keep in mind as you're designing and ultimately carrying these out and interpreting the results. So very similarly, talking about the three sort of phases, I'm going to start out with sort of the first phase, establishing a foundation, and the planning and funding phase. So the sort of tips and tricks I'd sort of give, especially for the people that are early on and trying to establish this line of research, is start out by building a credibility along the sort of line of research, establishing a foundation, and clearly establishing a great question. So going into a little bit more details about this. So building credibility. So in this idea, you want a line of research very similar to what Kurt and Connie just laid out for theirs. That's something that you can kind of carry forward over and over and over again. Surround yourself with similar people, similar experts that are also going to both build credibility to your trial as well as kind of carry it forward along with you. The foundation, this was really nicely touched on, but it's really important to consider, instead of going for large grants right away, you need to establish a foundation, particularly early on, particularly at your own center. Institutional grants are great for this. There's other opportunities through the REF and ASCS. But establishing a single center foundation, working through some of the kinks of actually carrying out the trial, and then ultimately helping to design your better questions once you expand it to a larger level. And then really it's about the question, and the question is where a lot of things get involved. And as Kurt kind of mentioned and Connie hinted at, if you don't involve your collaborators in asking the question, it's very difficult then to sort of gain a lot of the other aspects that we're going to sort of go through. So some of the pitfalls. Organization, clearly, and I think you've seen this in the other presentations, unrealistic goals and dealing with critics. And these are things that, especially when you're first starting out, that it's really easy to fall into. So from an organization standpoint, as both Connie and Kirk kind of mentioned, it's really important to have a point person, really important to have, or point people, from a regulatory standpoint, from a data organization standpoint, from a institutional regulatory standpoint, making sure that they are progressing, making sure you have a really nice and organized fashion. And then when you're setting your goals, you want them to be innovative. You want everybody to be motivated by them, but you also want to avoid unrealistic goals. This is the best way to burn out on a study, best way to decrease your ultimate enrollment and prevent you from reaching some of your goals, is to burn people out early on and have unrealistic early goals. And then critiques, this is something I think we all face. You submit a grant, you get it back, it's not accepted, and you go over and over again, or maybe it's a manuscript, early manuscript. Take these critiques, use these as constructive feedback, but do not get frustrated with them. Continue kind of pushing forward, especially if it's along this line of research that you sort of want to build your career about. All right, so the study execution part of this, and I think the theme from everybody that just talked before me, Kurt and Connie, it's about the team. It's about working together, creating this team, and motivating and ultimately carrying the team forward. And so in this phase, tips and tricks and pitfalls all kind of center around this idea. So maintaining engagement is a really important aspect. Milestones clearly is something that is important to both set and ultimately meet, and then making sure you have sort of study purity in a lot of respects with regards to your data, with regards to some of the regulatory aspects. So what I mean by engagement is as was kind of talked about both by the prior speakers, having regular meetings. We usually try to troubleshoot issues at least once a month with our trials. But we have touch points much more frequently, especially in the ones that are multi-center. We have you want to sort of remind of long-term goals. You want to remind people of long-term collaboration, but you also want to sort of motivate them and maybe even have them compete against each other or against other surgeons in the actual trial itself. The milestones are important, both to set realistic milestones, but also when you meet them to sort of celebrate that and to sort of meet as a group and kind of use that as a launching point towards trying to create the next milestone. And then the study purity idea is you want to make sure that as you're going, you're keeping track of patients, that you're keeping track of those that either you're losing the follow-up or some issues that are happening, and that you can sort of maintain this momentum without having it derail you. So a pitfall standpoint, I think accountability is something that's really relevant. Data breaches is something that I think you worry about, but a lot of the institutions have sort of have fail-safe mechanisms to work through. And then obviously the focus, and that I think that was the theme of almost every talk you've heard so far. So from an accountability standpoint, we try to send out this every Sunday night with all of our trials that are ongoing, all of the different investigators or different sites. And we have multiple different emails going out with the idea of trying to motivate people, trying to have them compete against each other in some respects, and kind of hold each site or each surgeon accountable. The data security, we use OneDrive. There's many different other institutional databases. RedCap's a wonderful way, especially from a multi-center standpoint, but it's really important to sort of have integrity to this, but also have security. And then the motivating and maintaining the focus. This is something that I found, and I see a lot of my colleagues that are younger, you sort of want to deviate, you want to maybe do sub-analysis, you want to do something that's detracting from the ultimate goal. So try to keep the big picture in mind, try to get to the finish line, and try to avoid some of these distractions. All right, so finally on the study completion aspect, as Kurt and Connie both mentioned, there's a lot of different definitions for this of how to get to the finish line and what that actually finish line means. So from my perspective, these are some of the tricks and keys, at least I've seen, to getting to that finish line and having some amazing products to look back on with pride. So authorship clarity clearly is something that both Kurt and Connie talked about. The message consensus, especially from the beginning, but carrying that through to the end, and then eventually really defining the writing plans. So just really quickly going through this, the authorship clarity is something you want to define from the beginning. You want to make sure people get credit for what they're putting into the study, so everybody is motivated, and you can organize it with regards to groups. You can organize it with regards to specific authors, but having that clear, very clearly defined right at the beginning is important. Messaging consensus is also important. Meeting as a group, identifying the message, being everybody agreeing on the message, the target for that message, where you're submitting them, where you're targeting. This is important both to carry forward the message and for the next step to be able to carry out these plans for different analyses that you potentially can do, different sub-analyses that you can do, different future goals that you can sort of take with the trial. As Connie kind of mentioned, these are really well done trial often has a lot of different avenues you can take it. And then from a pitfall standpoint, I think patience, defining roles, and study fatigue are all three things that I've seen and personally have experienced. From a patient standpoint, you have a set goal, you want to get to that goal, but sometimes you are having trouble with enrollment. Sometimes you're having trouble motivating investigators or surgeons to continue to enroll. But maintaining that motivation, maintaining that long-term goal of getting to the end is really important, especially to reward both you and everybody else for all the work you put in. defining clear roles when you are writing and when you are trying to do the analysis is really important, particularly when you have multiple manuscripts. And this also sort of helps to avoid that study fatigue. Sometimes at the end of a trial or end of multiple trials, you're sort of almost are ready to move on to the next or move on to something else. The manuscripts take out, maybe you get rejected from the first place you submit, but ensuring there's a timely and efficient completion of everything is really important. So sort of final thoughts, the themes from this, you know, you have a solid foundation as Connie and Kurt have elegantly demonstrated throughout their careers, and then being able to inspire and create a team that's going to carry it through and then carry you on to the next and the next, and producing the output that you can actually get to the finish line and actually make it worth everybody's time. So those have questions, reach out to me. This is my my contact information, and really appreciate the opportunity to be on this.
Great. Thank you, Ray. We got like 10, 12 minutes, I think, before we finish up here. So we'll see if you can put up slide number 6 or just list some general points for discussion. But Connie, one important point you made was identifying and establishing a clinical coordinator and assembling the team. Obviously critically important. How do you do that? I'm sure it takes years to really establish your research team. You know, what were the pitfalls you ran into in trying to accumulate a team that could really be on the same page and help you with your research?
Sure. So kind of thinking back early on, um, uh, trying to do things yourself when you're trying to practice because you don't really have, you know, that person. The big benefit of that is that you learn some of those steps, but really it's important to kind of establish that if you're going to recruit patients and do quality, compliant clinical research, you need to have someone dedicated to that role. And so clinical coordinators, certainly where I am, are extraordinarily costly, but motivated medical students, early on research assistants who were interested in medical school. So maybe they were partially doing work in the laboratory as well. They would start to pick up on some of those tasks. So really, students, fellows, and where I was at the University of Pittsburgh. We had research residents who did a full year of research. And so you do need that human resource. And if you get people engaged, they actually want to find the answer, and they will go out of their way to try to retain the subjects, get them in to do the research tasks, and also hand off to some of the research, other members of other people's research teams working together. You know, if it's a give and take, it works. It can work very well.
Yeah, great. That's helpful. I think the clinical coordinator is so critically important. And one thing I've learned here at HSS is certainly having students involved in things is great, but as research becomes more complex and really doing higher level intervention trials, ultimately we need to establish a career pathway, a career ladder, so you can retain your clinical coordinators, because someone who leaves after a year or two, you lose their institutional memory. So ideally, we're trying to put in place a career ladder where it's really seen as a career pathway with advancement, salary, the support that goes with that, increasing authority to really be able to retain good people. Kurt, I made a comment earlier about the challenges with recruitment in studies. What challenges have you faced as far as getting your colleagues involved? It's clearly we all are a big institution with a lot of volume, but how do you really get your colleagues, your clinical colleagues, to be involved in the middle of a busy clinical practice? They're seeing patients in the office, they're busy, may not always be top of mind for our colleagues as much as it is for you and I. Any thoughts on how we engage our colleagues to help us with enrollment?
I think what Eric showed is that if you get them to agree to participate, and you're monitoring you have coordinators monitoring the eligible patients that come through their clinic. You just show them their enrollment, and they don't like to lose because they're competitive. And so and that's just, they don't like to lose. So basically they see that they certainly don't like to lose to me, and they have more volumes than me now that I'm older. And so you just have a good competition to see who enrolls the most patients.
Yeah, awesome. It's good motivation. I agree. I agree. You also made an interesting point about single site versus multi-site studies, which I think is a very important point there. You know, everybody holds up the multi-site studies as the gold standard, and it is, but they're more challenging. But I think it's an important point to first establish efficacy or some early data in your single site study, which also allows you to really power a larger study, right?
Yeah, I think you really do need that, because in a single site you have better control. You usually have the inventor or the person most engaged in it. That's whether it's a novel device, technique, or whether, and so they're doing it under ideal conditions. And so if it doesn't work there, then or if it doesn't have a good signal there, the signal will get blended out, or the effect will probably lose it as you try to show the same techniques and protocols across different centers.
Yeah, sure. Eric, you have an impressive experience already in your early career already of having done a number of clinical trials. Are randomized trials truly the gold standard? And what are your thoughts regarding prospective longitudinal cohorts that may be more realistic or a little easier, if you will, to do?
Yeah, I mean, without question, like a couple points. One real quick, just involving when you first are starting out and how to do this. You know, you mentioned the clinical research coordinator, which without question is important. I do think Connie's point with research assistants, we've done some value analyses on these, and you know, research is about a third the price to start out with. And so when you're gauging your colleagues, we actually engage one of our private practice groups here in Atlanta pretty frequently in most of our trials, and having research assistants that can sort of reach them or involve them, and they even at this point have one of their own, is really helpful. And then a coordinator kind of overlooking that. But yeah, as far as like randomized controlled trials versus prospective cohorts, you know, RCTs are great for specific questions, but as I kind of hinted at early on, if you look at the fragility of a lot of randomized controlled trials, it's actually pretty impressive. And so, you know, it's hard to execute, it's hard to care, it's hard to finish, and especially in America, it's hard to get patients to enroll. And so there's a lot of effort, and then sometimes the outcomes are pretty fragile. I think the advantage of these prospective observational studies, or these prospective cohorts, with you have a bunch of interested, similarly motivated, similarly minded people, you can answer some really important and maybe multiple really important questions. And although it's not technically the quote unquote gold standard, it's as I mean, Connie and Kern can talk about and show, you can answer some questions that's honestly like a RCT would have a lot of difficulty answering. So truthfully, like that is probably the future, and that's probably something that's you can see trending just in general at our society and other societies. But I do think there is a role for specific questions that an RCT probably is better suited for.
Yeah, I think it's probably no one size fits all. It may depend on study question, the population, you know, any of a number of factors. Because, you know, like randomized trials for surgery can be challenging. You know,
100%.
Connie, back to the issue of your team and clinical coordinator, the other important or critically important part is biostats. I've made the mistake for years of, you know, you carry out this plan study, you do the study, you collect the data, and then you consult the biostatistician. Well, I've learned that's not the right way to do it. You should have the biostatistician involved from the outset. What resources do you have at Stanford? What challenges have you seen as far as getting biostats involved? Not just for planning the statistics, that's just the small statistics part, but broader study methodology.
Yeah, so that's an area where we're actually very fortunate. Our department chair has invested in biostatistics, and we have a biostatistic core led by Jamie Koltzoff, who trained at HSS, and we've now expanded where she has assistants. And we stress to all the residents and all the fellows, do that biostatistics consult first. And so in that sense, at both institutions I've been at, there has been biostatistics support. So very fortunate.
Good point. And then the other issue that come up, Bob, a little bit, you and Eric, you touched on authorship. Critically important that everybody gets credit. Curry made good points about, you the PI shouldn't be the lead author on every study. What challenges have you seen as far as authorship? And importantly, or most specifically, do you fill out an authorship, a planned authorship form at the outset of a study, understanding that things change over the years it takes to do a study? But how do you address authorship issues? And because it can become thorny issues. Eric, maybe you can comment from your standpoint.
Yeah, so from my standpoint, so I think a plan initially, define roles. We have clearly, when we approach a single or multi-center study, we have clearly defined sort of definitions of what actually constitutes an author with regards to participation efforts. We also have clearly defined sort of goals with regards to is it going to be a group? Is it going to be, how are we going to deal with that before we start the study, when we're in the planning phases, before we even start approaching some of the funding mechanisms. But at least my experience has been that is a fluid mechanism as it goes. And then when you get to that completion standpoint, it's important to give everybody credit, but it's also important to evaluate as you go and as you start to plan out writing different manuscripts, depending on the phase that you are at. I think it's important to sort of as a consensus or as a group come together and maybe reevaluate some of the initial goals for authorship. But it's really clear before you start writing to make sure everybody is on the same page. And then the messaging consensus, as Kirk kind of mentioned, I actually find that you'd think it'd all be the same, but you'll have the same analysis, everybody's reading the same paper, the same analysis, same abstract. And you have people disagreeing on how we should interpret that or how we should approach it. Or as Kirk kind of mentioned, like what are the number of manuscripts that we're going to write with this. And so it's having consensus is really important, especially if you're going to maintain momentum, can kind of continue moving forward, which I can tell you at my phase, that's like one of the most important aspects is getting to that next phase and next phase and next phase. And that's probably just as important, at least in my mind, as the actual authorship. So.
Yeah, thank you. And last question I'll ask maybe Connie and Kirk just very briefly, just comment on, probably the vast majority of our surgeons are in private practice settings where they may not have all of the resources and infrastructure for these more complex trials and coordinators and things like that. And so what it's challenging, but how might we get our private practitioner colleagues involved in randomized trials?
Yeah, so I think the first crawl step is to start tracking your own outcomes. And there are new software packages that actually integrate with the medical record and allow this to be done much easier and potentially consider if it's an area that the AAOS has a registry, and that can get people started.
Sure. Yeah, that's helpful.
I think that, I think they can participate in prospective cohorts, particularly when they have to get a patient-reported outcome, and then they can follow that. The follow-up can be from a central site, so that's sort of the first step. But if they really want to participate in randomized trials, somehow they need to get a research coordinator, because someone needs to consent and talk to the patient. That's not practical, even for us to sit and spend 15 or 20 minutes with the patient in order to do that. So they would have to make an investment in that. And I think some certainly will, and some may not. I guess it depends on the volume and everything.
Sure. No, thank you. Well, you've all hit on very important points. It's been very instructional. I always learn a lot from all of you and all of my colleagues. And so with that, we'll probably wrap up. We're at an hour. Again, we should thank Smith and Nephew and recognize them for their nice support here. Thank you all very much for your participation. Steve, any final thoughts? Or Peter?
Oh, yeah, thank you very much. Amazing talks. I learned a lot. I have one question. Let's say we have a private practitioner who has an amazing idea, find the funding through industry, but they don't really know how to start. To get a certified coordinator is expensive. Statistician may be difficult to talk to. And one thing which I usually find from our fellows who graduate and leave and call me later what to do, how to negotiate a contract. Do we need to have a lawyer with a CTA? How to negotiate a contract with a hospital? What is your experience? What would be the one thing you would tell them to do? Just try to figure it out themselves, get a general counsel, contact a friend? I don't know. because I get that question quite a bit, and we have a system established, so I know what I would do, but I don't really know what would I do if I were a young practitioner who's starting practice.
Yeah, my sense would be to try to hook up with individuals at an institution where they have some of that research infrastructure. Because there's contracting, budgeting, legal review, material transfer agreements, potentially, CTA, clinical trial agreements. All those things are important, and it becomes more complex at these higher level trials. And I would be careful doing that on your own. So that's the challenge of how do we get our private practitioner colleagues involved? Maybe we partner with them so we can help use the resources of our institutions, the infrastructure that's set up at Stanford or Cleveland Clinic or Emory, yet to help our colleagues.
Agree.
Partnerships. It's all back to teamwork, partnerships. That's where societies, or the ASCS or AOSSM, Sports Medicine Society, can help hopefully bring people together. So, great points.
Thank you.
Thank you. Steve, any final words? If not, we'll wrap.
No, I'm good. Thank you all for joining.
My pleasure.
All right.
Everyone have a good night. Thank you all.
Thanks, everybody.
Thanks,
Good night. Thank you.
everybody. Learned a lot.
Bye-bye. Thank you.
Take care.